Hi, in this video I'm going to demonstrate how to determine ABO blood groups. Before we can start, we need to have a few things ready. 145 millimolar sodium chloride, blood samples, and blood typing antibodies. So for this first sample I will label 5 microcentrifuge tubes, one for each antibody, and a negative control. We're going to test our blood against anti-A, anti-B, anti-AB and anti-rhesus D antibodies. If you want to catch up on the theory for this lab, you can check out the video lecture. You can also take a look at the second video I made about osmotic fragility and hemolysis. Once we've labeled the tubes, we will dilute the blood in sodium chloride. So I will add a few drops of 145 millimolar sodium chloride first. It's important that it's 145 millimolar, since that is the full salt concentration. If the salt concentration is too far off, it will lead to an osmotic shock and burst the cells. You could use a real pipette to add the exact volumes, but this is a qualitative analysis and it'll work just the same using these droppers. Alright, you see the volumes aren't exactly even, but that's okay. Now we can add a single drop of blood to each tube. It can be a little tricky to get the drop to hit the bottom of the tube. That's fine, since we're going to mix the contents anyway. Once we've added the blood, we give each tube a flick or two and make sure the solution is nicely homogeneous. Now it's time to add the antibodies, starting with anti-A. It's more than enough to add a single drop. Remember how the blood groups are defined by the antigens on the surface of the red blood cells. The antigens in the ABO system being carbohydrates attached to proteins and lipids on the cell membrane. We additionally test whether the sample contains a protein called the rhesus D factor. The antibody we are adding is specific for a particular antigen. So, the anti-A antibody binds only to the A antigen, and anti-B only binds to the B antigen. To test for the presence of both at the same time, we use a mix of anti-A and anti-B. If an antibody finds a matching antigen on the cell surface, they form a complex that binds many cells together, leading to the formation of a clump. This process is called agglutination. Once we've added all the antibodies, we mix the blood and antibodies and wait for about 10 minutes before we can see if a reaction has occurred. Finally, we can have a look at our results. For each antibody, we check if we can observe any agglutination. We can see that there are clumps for both anti-B and anti-AB, and that there is neither agglutination in the negative control, which is good, nor in the tubes with anti-A or anti-D. From this we can infer that the donor was type B negative. And if we go and check the label on the blood sample, we can confirm B rhesus D negative. You can see on the tube stem that I've already prepared a second sample, which we'll take a look at right now. Okay, here's our second sample, and it looks a bit different than the first. We have agglutination everywhere except in the negative control. That means the cells in this sample have all three antigens, A, B and D. Therefore the donor was AB positive. Now I'm going to repeat that for our third and last sample. And there's the third sample. This time we've only got agglutination against anti-D, so this donor was type O positive. We can check again on the blood tube, and indeed, O positive.